Welcome back. This is Lecture 30, Coliform and E. coli testing. Think about the logic we've used so far. Lecture 28 asked how many organisms are present, total. Lecture 27 asked whether one specific named pathogen is present. Today introduces a third logic entirely, one we haven't used yet in this level, the indicator organism. It's a stand-in signal for contamination you can't see directly, and by the end of this lecture, you'll understand exactly why that reasoning works, and just as importantly, exactly where it doesn't. Here is what we will cover today. Number 1, you will understand the indicator organism concept, why a harmless organism can flag the possible presence of dangerous ones. Number 2, you will know the three-tier hierarchy, total coliforms, fecal coliforms, and E. coli, and why specificity increases at each level. Number 3, you will be able to define a coliform biochemically and explain what the 44.5 degrees Celsius fecal coliform test actually proves. Number 4, you will understand how the mug test detects C. coli through beta-glucuronidase activity, and how to read a fluorescence result correctly. Number 5, you will know the critical exception, why E. coli 0157H7 can escape standard coliform and mug-based testing entirely. Let's start with the core concept. What is an indicator organism? Lecture 28 asked. How many organisms total? Lecture 27 asked. Is this one specific pathogen present? Today introduces a third question entirely. Does this organism's presence suggest a pathogen might be here too, even if we haven't tested for it directly? Here's the logic. Coliform bacteria, and E. coli specifically, share a habitat with dangerous pathogens like Salmonella and Shigella, the intestinal tract. They are far more abundant than those pathogens, and far easier to test for. If they're present, that tells you fecal contamination has occurred, and pathogens could plausibly have traveled that same path. But here's the limitation. And I want to be precise about this. An indicator is a probability signal, not a direct test. A negative result lowers the likelihood of fecal contamination, it does not prove a pathogen's absence. A positive result raises suspicion, it does not confirm a pathogen is actually there. Think of it like a smoke detector. It doesn't detect fire directly, it detects a reliable byproduct of fire. Coliform testing works exactly the same way, using an organism that's easy to detect as a proxy for a risk that's much harder to detect directly. Not all coliforms mean the same thing, and this is important. As you narrow the definition, the organism becomes a better, more specific indicator of actual fecal contamination. Look at the three tiers on this slide. Total coliforms, the broadest tier. This includes environmental species found in soil, on plants, in water generally. It's the weakest indicator, because presence alone doesn't confirm a fecal origin at all. Fecal coliforms, a thermotolerant subset. These specifically grow and ferment lactose at 44.5 degrees Celsius, a temperature that favors organisms adapted to the warm-blooded gut specifically. That elevated temperature is what makes this tier meaningfully more fecal-specific than total coliforms. And E. coli, the narrowest and strongest tier. E. coli is not free-living outside the gut, which makes it the best single indicator of fecal contamination currently available. As you move down this hierarchy, you're trading breadth for specificity. Total coliforms catch a lot, including plenty of false alarms. E. coli catches less, but when it's there, you can trust what it's telling you. Let's define exactly what counts as a coliform, because this is a functional definition, not a taxonomic one. Coliforms are gram-negative, facultatively anaerobic, non-spore-forming rods that ferment lactose with both acid and gas production within 48 hours at 35 degrees Celsius. That's the actual working definition. Let's break that down. Gram-negative rods. This connects directly back to Lecture 26. Like every organism in the Big Six, coliforms are gram-negative. Facultatively anaerobic, they can grow with or without oxygen, which happens to be exactly the environment inside a gut. They ferment lactose, acid and visible gas within 48 hours at 35 degrees Celsius, and this fermentation reaction is the actual basis of every classical coliform test you'll encounter. And the genera included: Escherichia, which is of intestinal origin, plus Enterobacter, Klebsiella, and Citrobacter, which are often environmental or plant-associated in origin. Notice that last point carefully. Several coliform genera are perfectly normal to find in soil and on plants. That is exactly why total coliforms alone is such a weak indicator on its own. It simply cannot distinguish ordinary environmental presence from actual fecal contamination. Let's look at the toolkit for different ways to actually detect coliforms: any coli, MPN, or multiple tube method. Sample is diluted across replicate tubes, and the resulting pattern of positive and negative tubes gets matched against a statistical table. This gives you a statistical estimate, not a direct count. Membrane filtration. Sample is filtered through a membrane, and that membrane is placed on selective agar, with colonies counted directly. This is a direct count, and it's more precise than MPN, chromogenic, or mug testing. Enzyme substrate media change color or fluoresce when specific bacterial enzymes are present. This gives you a presence or absence result, often available the same day. And Petrifilm, or dry film methods. Pre-prepared selective media on a ready-to-use film, sample added directly, no plate pouring needed. This gives a direct count with fast setup. The next two slides go deep on the mug test specifically, because it's the method you're most likely to encounter on a modern QC bench, and it has a critical blind spot that's genuinely worth understanding in detail. Let's talk about how the mug test actually works, mechanistically. About 95 to 97% of E. coli strains produce an enzyme called beta-glucuronidase, often abbreviated GUD. Mug testing exploits that enzyme directly. Follow the reaction on this slide. First, mug substrate is added to the growth medium. On its own, it's colorless and non-fluorescent. Second, if E. coli is present, its beta-glucuronidase enzyme cleaves the mug molecule. Third, that cleavage releases a compound called 4-methylumbiliferone, which glows blue-white under long-wave UV light, specifically at 365 nanometers. Here's how you actually read the result. Hold the sample under long-wave UV light. Blue-white fluorescence is a positive result for E. coli. No fluorescence is a negative result. This method is fast, often same day, and far less labor-intensive than classical fermentation tube methods. But I want you to notice something in what I just said. 95 to 97% is not 100%. The next slide covers exactly which strain falls into that remaining gap, and why it matters enormously. Here's the critical exception, and this is genuinely one of the most important things in this entire lecture. Recall Lecture 27's SDC entry. E. coli 0157, H7 is one of the big six. It is also, in most strains, beta-glucuronidase negative. That means it typically will not fluoresce on a standard mug test, even when it's actually present in the sample. There's a second blind spot too. Most 0157, H7 strains also grow poorly at 44.5 degrees Celsius, the standard fecal coliform test temperature. So that pathway can miss it as well. Why does this happen at all? Indicator tests were never designed to catch every pathogenic strain specifically. They test for a trait that's common to E. coli generally. And 0157, H7 simply happens to be an outlier on that particular trait. What does this mean in practice? A negative mug result, or a negative fecal coliform result, does not rule out 0157, H7. Products with elevated SDC risk need targeted testing, the method we covered back in Lecture 27, not a coliform proxy standing in for it. This is exactly why indicator organisms and targeted pathogen testing are complementary tools, not interchangeable ones. Each one covers a gap that the other one has. Now let's look at what this actually looks like in practice. Take a look at the three photos on this slide. First, mug fluorescence under UV, a tube or plate glowing blue-white, showing a positive E. coli result. Second, a Petrifilm E. coli and coliform plate. Red colonies with visible gas bubbles indicate coliforms, and blue colonies indicate E. coli specifically. And third, Empyrean fermentation tubes, a rack of lauryl tryptose broth tubes showing gas production in the small inverted Durham tubes, a presumptive positive result. Before we close, let's walk through where coliform testing commonly goes wrong. The first mistake is treating total coliforms as proof of fecal contamination. Total coliforms include common environmental and plant-associated species. Only fecal coliforms or E. coli specifically confirm an actual fecal source. The second mistake is assuming a negative mug test clears a product of E. coli O157H7. Most O157H7 strains are beta-glucuronidase negative. A negative mug result simply does not rule this specific pathogen out. The third mistake is treating an indicator result as a pathogen confirmation. A positive coliform or E. coli result raises suspicion, it does not confirm that any specific pathogen is actually present. That still requires targeted testing. The fourth mistake is confusing MPN with a direct colony count. MPN is a statistical estimate derived from a pattern of positive and negative tubes, not an exact count the way a plate count is. And the fifth mistake is ignoring the 44.5 degrees Celsius requirement for fecal coliform testing. Running that step at the wrong temperature defeats its entire purpose, since that elevated temperature is precisely what makes the test more fecal-specific in the first place. Let's bring this lecture to a close with the key takeaways. Number 1. An indicator organism is a probability signal, not a direct pathogen test, a stand-in that's abundant and easy to detect. Number 2. Specificity increases down the hierarchy. Total coliforms are the weakest, fecal coliforms are better, and E. coli is the strongest, single best indicator available. Number 3. Coliforms are defined functionally. Gram-negative rods that ferment lactose with acid and gas within 48 hours at 35 degrees Celsius. Number 4. The mug test detects beta-glucuronidase activity. A positive E. coli result glows blue-white under UV light, and it's fast and reliable most of the time. Number 5. E. coli O157H7 is typically beta-glucuronidase negative, which means it can escape both mug testing and standard fecal coliform temperatures. Number 6. Indicator testing and targeted pathogen testing are complementary. Neither one replaces the other. That is Lecture 30 complete. You now understand indicator logic. You know why coliforms and E. coli function as proxies for fecal contamination, how the testing hierarchy narrows in specificity as you move through it, and exactly where that logic has a real, well-documented blind spot. Up next is Lecture 31, Salmonella Detection, Classical and Molecular Methods. That lecture takes us back to targeted pathogen testing, the kind of testing that exists specifically to catch what indicator organisms like the ones we covered today cannot. Take a look at the roadmap on this slide. Lectures 24 through 29 are done. We are wrapping up Lecture 30 right now, and 5 more lectures remain in this level. If this lecture helped you, please give it a like. Subscribe for new lectures every week. And drop your questions in the comments, they get read and answered. See you in Lecture 31.